Good morning, everyone. This is my last week in plant molecular biology lab at Koç University. So I decided to record my last two experiments. The first experiment is Western Blot. This is the second day, so I did overnight. What was it called? Overnight transfer of proteins to the membrane. And now let's get rid of these ice packs, and I will need this container. I worked in this lab in my undergrad years, and now I got accepted from Göttingen University at Germany, so I will be leaving in the upcoming weeks. I will move to Germany and start my PhD studies. I am so excited about that. Everything is ready. The only thing I am waiting is my visa, and hopefully I can get it as soon as possible, because it's really hard to wait when everything is ready. I'm really curious about the life in Germany and how the research goes in that country. Okay, let's put the transfer buffer in the fridge first. My protocol in here. So first I will rinse the blood with water and then dye it with Poncio to see whether transfer is successful or not. This is the membrane. I am taking out the filter papers first, and let's see whether the leather is transferred or not. Yeah, as you can see, the leather is transferred, and now hopefully there is no protein in the gel. I will put it in here. Now I will get some water and rinse the blood to get rid of transfer buffer. I think 5 minutes is done, and still I don't see any bends, but let's give it a minute or so. I have my lab coat on. And by the way, this is my name. My name is Deren. Nice to meet you. Oh, there is no bands, as you can see, but I have hope still. Now I will wash the membrane with TBSD and then go for blocking. I love you, baby, and if it's quite alright, I need you, baby. Membrane will incubate for an hour in the room at the room temperature with blocking buffer, and after that I will continue with the primary antibody incubation. I was in the 3rd grade when I first started working in this research lab, and it was a huge milestone for me because I wanted to work in a research lab since I was a kid. I was very enthusiastic about like doing research and being in the lab, and because of the COVID, the university and all the things were online, so even I wasn't able to take my lab courses one-to-one, like in person. But when the school starts, they did some workshop for the lab courses that we missed, but even that it was not enough for me. So I started to contact with my professors, and while taking a lab course, meanwhile, I see that I don't want to work with cell cultures. Definitely, I don't want to work with mice. And at the end, the only thing left in Koç University for me to work with was plants. And I was really happy about that because I love plants. I always loved plants in my whole life. So it was a great opportunity for me to work with plants. And the thing is, the PI of this lab came to the school at the same time with me. I came to Koç University in the 2nd grade, and the professor came to the university at the same time with me. And while all the things were online, the lab was like growing, and he was hiring PhD and master students, and also some undergrads started working in the lab. And when all things are done, I've started the lab, so I'm lucky about that. And all the people are very helpful in the lab, so they taught me a lot, and I appreciate all of them. And I was also so excited about working in the lab, and I like took all of the information in an instant moment. so it was easy for me to learn all the techniques and understand the theoretical background of the experiments. So overall, it was a wonderful opportunity for me to work many projects and experience variety of methods in molecular biology. Blocking is over. Now I will incubate the membrane with primary antibody, but first I need to prepare the antibody. Because the proteins are expressed with flag, I am using flag antibody, and the ratio is 1 to 1000. Therefore, I will put 3 milliliters of blocking buffer and then 3 microliters of the antibody. After primary antibody incubation, I washed 5 times. The membrane is washed 5 times, and now I will prepare the secondary antibody. After the washing steps, the membrane will be ready for lycor. Ta-da! All of the antibody incubations and washing steps are over. Now I will use lycor to see do I have any protein attached to the membrane. I have vermiculite on the membrane again.
Yeah,
like,
why?
Is it my curse or something?
I have no idea. Last time, I have a fly in my membrane.
Fly?
Fly. It's a tiny fly. It's
Okay.
like a trauma for me.
It's the like nirvana. I'm so excited. This is my last Western Blot.
Aww.
Okay, we are ready. And we can start the scan. This time my membrane is big.
How many samples?
7.
Whoa.
Because normally I have 4 samples, 4 new samples, but I want to just load my old Medicago samples to check. Maybe magically
expect?
something happens. Okay. Stop.
There's something inside there, but no, it's not.
What are those?
I have no idea, but don't be afraid.